Hello, hello. I'm turning on my Restream chat so I'll be able to talk to everybody while you're popping up on here. I'll give everybody a minute to hop on here and say hi. Restream takes forever sometimes to load. That's okay. Whoop. I clicked the wrong thing. Hello, everyone. I am hanging out here a day late on my show. So today I'm going to talk about Today I'm going to talk about gene editing. A day and an hour later than I normally do because I wasn't feeling so well yesterday. But it's better today. Hugs to you too. So today we're gonna talk about gene editing. Gene editing is a fun thing, and we're primarily gonna focus on CRISPR. Hello everyone. So if you're not familiar with this type of format, what I do is, this was a voted on for topic, and it was tied last week with the Big Bang Theory. Not the TV show, the actual theory and the origin of the universe. So, okay, I hope you can stay for it too. It's not a very long episode because it's fairly new technology for the most part. And in regards to gene editing, it's really exciting stuff. So this was tied with the topic of the Big Bang Theory, so that's why I'm going with this today for this episode. So let's go ahead and get started. So that's one of my cool intros that Gonna Go For It is made for me. So he's pretty cool, and he makes cool stuff like that. So let's go ahead and get started with this gene editing stuff. So pretty much what I'm going to cover today, what I want to talk about is, how does it work? What is it? How can it help us? And how far along are we from implementing this in like actual people and stuff? So it's pretty exciting things going on with this CRISPR stuff. So what I want to do is talk a little bit about what it is and how it works, and then we'll move on to that other stuff in just a bit, because it's kind of important that you get an idea of how it works and that sort of thing. So CRISPR, what does CRISPR stand for? Well, it is clustered, regularly interspaced, short palindromic repeats. So that's a mouthful, isn't it? But that's essentially what it is. Oftentimes people don't think, they think, okay, well, technology, it's got to be a machine or something. It's got to be some kind of apparatus. Well, this is all biological. It's exclusively a biological mechanism. So while some do think of technology as a machine, CRISPR is all molecular biology. So that makes it inherently really, really cool, because there's not a whole lot of actual tech type of stuff associated with it. All the hard, heavy lifting is done on the molecular level. So what we're going to do now, I'm going to kind of break down some of this information for you. Essentially, CRISPR, it's a tool developed from bacterial CRISPR, associated protein 9 nuclease. Okay, so CRISPR is actually a thing that's found in bacteria. So when we say CRISPR associated protein 9 nuclease, you might have seen it in various magazines or articles or on the news as Cas9, Cas9. Well, in case you're wondering and you're like, that was a big mouthful, what the heck do all those words mean? Well, a nuclease is an enzyme that kind of chops up genetic code into smaller bits. And so CRISPR kind of goes through Cas9 and chops up these different parts of genetic code into bite-sized portions, right? So why is Cas9 CRISPR important in bacteria? Well, the cool thing about it is a function of what we would call adaptive immunity that we can find in specific types of bacteria. Now, what this type of biological mechanisms allow is it allows bacteria to quickly respond to and completely eliminate foreign genetic material. So we often see this type of response in regards to viruses trying to infect bacteria. There have been 3 types of CRISPR mechanisms that have interested scientists in regards to genetic engineering research. But the second mechanism is the one of most interest due to it only needing one type of CS9 to silence genes, as well as only 2 types of RNA. Now, RNA is kind of important. Hello! RNA is kind of important with this because these 2 types of RNA attach to the CS9 in order to make this enzymatic complex. So that sounds really fancy, in order for it to function properly. Certain enzymes require things to attach to them in order for them to even do their job. Okay, so these types of RNA, there's only 2 of them in the CRISPR-2 mechanism. That's what we're going to talk about today. I'm not going to get into the others. So we're just going to talk about the one that scientists are like, oh my gosh, this is what we need. 2 types of RNA attached to the CSA9, and that's what gets it to go. So I'm going to show you a little picture of that so you can kind of have a better look at what this kind of looks like. So, all right, so here, if you're not familiar with RNA, RNA is a type of ribonucleic acid. It's type of genetic material. All right, and so the 2 types of RNA that we're talking about are the CRISPR RNA, that's the cRNA, that's over in that picture, the cRNA, and then the other one is called trans-activating RNA, or it's also called the tracer RNA. So these 2 types of RNA, and we see in our little purple bubble there, that's our Cas9 enzyme. And we have our cRNA, that's attached to the Cas9. And then that tracer RNA attaches to that cRNA, and that gets the Cas9 to go, okay, we got to snip this. Now, what makes the cRNA important is that it attaches to your genetic code of interest. And so once it attaches to that DNA that you want to cut out, that tracer RNA attaches to it and to the cRNA and goes, okay, Cas9, we found the genes we want to delete and remove. We can cut them now. Here it is. And that's where Cas9 goes through and starts snipping. It's pretty cool stuff. But it can get, oh my gosh, kind of crazy. Now that NGG sequence that they have in there, that's just like a recognition portion of where the Cas9 kind of attaches to the genetic code. So I hope I'm not losing anybody right now. Sometimes this stuff can get pretty heavy and you're like, wow, man, that's a lot of words. So I'm going to give you a little bit of background, all right? So I might say their names wrong. I'm not very good with names sometimes, and I kind of stink at that, so I'm just saying that in general. Doudna and Charpentier labs first harnessed the ability to gene edit through the CRISPR-2 mechanism. So what I just showed you in the previous picture was the CRISPR-2 mechanism of the 2 RNAs and the CS9 attaching to the genetic code and cutting it out, right? So they took Cas9, and they synthetically made what we call a single guide RNA, okay? And it acts like those 2 CRISPR and trans-activating RNA, but combined. So they just made one RNA that can attach to the Cas9, and so it kind of eliminates all of those other steps of the CRISPR RNA finding the DNA, and then the trans-activating one attaching to the CRISPR RNA, and then it cuts it. We got rid of all those steps. So they's like, we're going to get rid of those. We're just going to use this synthetically made one called single guide RNA. So they found that they were able to edit genes by removing code and adding donor DNA. So not only could they remove genes, they could introduce new genetic code into those DNA strands. I know, it's fascinating, isn't it? So if you have questions or if I'm going too fast, let me know and I will slow down. Now, there have been, get this, so this is how far science has come. Now, they went through 3 different mechanisms trying to figure out which CRISPR mechanism was the best. We said the second one, right? It takes a couple of RNAs in the CS9 to make this super complex in order to nom on the genetic code, right? Scientists are like, we're going to get rid of those 2 RNAs and make our own, so that makes it more efficient. Well, now, scientists have actually modified the CAS9 into 3 different types for it to do different things. Hello! So the 3 different types of CAS9, you have the natural one, what they call the wild type that you find in bacteria. It can snip the double-stranded DNA and activate that break and repair the DNA. All right, so you have the naturally found one, the wild type CAS9. Now you have a mutated one that they've done where they're able to use 2 guiding RNAs can be introduced to 2 in different opposite positions and opposite double-stranded breaks, so you can do multiple repairs on the same DNA with the mutated CAS9. Then there's another one where they've gotten rid of the function to where CAS9 cuts up DNA, and instead they can put other things on it to get it to deliver different things to the genetic code. Why? Who do I have to speak to take my DNA out and replace it with Brad Pitt? We will talk about all of the different challenges that we have to overcome before we get to people. Can you create a new beast? There's an interesting discovery that's come out in 2017, and I'm going to talk about that in just a minute, about making new semi-synthetic organisms. And so the last type of Cas9, what you can do is you can have effector compounds that allow the Cas9 to attach to a specific point on the DNA. You can, for example, initiate protein making. That's transcription. So you can activate certain points of the DNA to make proteins associated with a gene. You can stop gene expression so that Cas9 can snap onto that DNA and just say, nope, we're not gonna transcribe this section of DNA. We're not gonna make any proteins here. Or you can actually add something like GFP. That's a fluorescent dye. Attach it to Cas9 and light up the type of genetic code that you're hoping you can see underneath a microscope. Sometimes if you have a powerful enough microscope, you can have a fluorescent microscope and be able to see that sort of thing. It's alive, it's alive, that's right, isn't that cool? So I'm going to show you again a bit how CRISPR works without freaking everybody out going, this is so much, oh my gosh. All right, so here I have a picture. And here I have the Cas9 protein. So in the top bubble here, I'll use my little pointer here. Here we have our Cas9 protein, and inside we have that single guide RNA. I do love science and I work in science, so I'm in a STEM-based field, so it's fun. This is something that I do to try to contribute and give back to the community. So, so here within our Cas9, that's what this is. Here's our nuclease, our fancy CRISPR enzyme, and it's going to carry this RNA. This particular RNA is our guide RNA. And then what it's going to do, and this is going to be kind of complementary to the stuff that we want to get rid of, all right? So once they come together and form their little complex, they travel over to the genetic code, right? They're like, wee, and they go to the genetic code, and then it finds the part of the DNA that's associated. So that particular guide RNA says, hey, here it is. Here's our gene. See, I can attach to it. And so then that's what signals the Cas9 to cut. And so that's what it does. It does what we call cleave. It cleaves at the site, and you can introduce new DNA to fit right into that spot. So that's kind of, in a nutshell, how CRISPR works with CAS9, that particular enzyme. Is this live? Yes, I'm live talking to you now. But if you're, can you do this in living things? You absolutely can, and the living things can survive. Isn't that cool? Science does absolutely fascinating and wonderful things, huh? So, let me see. All right, here we go. So the next thing that I want to do, I want to show you actual footage. So here we have a picture, and this, oh gosh, this is just absolutely fascinating. We have here CAS9, that's labeled there, and the RNA. Oh, you're following me now, yay! This is real space and real-time dynamics with the CRISPR-Cas9. Now, how they managed to do this is through something called high-speed atomic force microscopy. So microscopy just means microscopes. And so they have this really super fast atomic force type of microscope. It's where it's able to line me with science. So where it's able to take this tiny little needle, and it consistently moves back and forth at a high speed in order to make these images. And so these images they can put together kind of like stop motion animation in order to see how the CS9 breaks apart like it just did now. CAS9 and RNA breaks apart the DNA. This just came out, I believe, a few weeks ago. And this comes to us from a team from Kanazawa University and the University of Tokyo in Japan, who managed to publish this paper in Nature. And the name of the paper is Real Space and Real-Time Dynamics of CRISPR-Cas9 Visualized by High-Speed Atomic Force Microscopy. If only all teachers were like you. I used to be a teacher for about 4 years. It was kind of fun. Yes, I watched Sam Harris's podcast with Jennifer A. Doudna about CRISPR and was blown away. It's very interesting. Yes, it is very interesting. So this is an example of what happens with CRISPR. This is real time, and we're able to actually see what shape the enzyme is, relatively speaking, and to actually watch it break apart. How can you CRISPR enough of them to make any difference? That is a good question as well. So one thing that I do want to show you, I'm going to talk a little bit about the challenges that are associated with CRISPR technology. And so what I have here, can you nanofix badly failing organs? If it's with CRISPR, we're still a long ways away from stuff like that. It's much easier to work with, for instance, embryos. And so I'm going to talk a bit about what scientists managed to do with an embryo just this year. CRISPR's 2017 achievements, as we call it. So here I have a GIF image. And if you're curious and you want to read more about this, at the end of all my episodes, I have all of my sources listed. And if you want to, and I do update my slides on scientistmel.com, that's scientistmel, just like here, .com, and you can download my PowerPoints, dig into them, and investigate all of my sources as well and learn more about it. So fear not, you can check them out there too. All right, so what I have here in this particular image, this GIF image, is an example of CAS9 actually removing viral DNA. And so kind of the mechanisms behind it. Now, it doesn't actually look like this. It's not like a big old blob of bubble gum as we saw in the previous picture, but it's still kind of a general idea of what we do. So what I want to do now is kind of show you all of the achievements associated with CRISPR in just this year. So I'm going to go to my big monitor here and kind of get that thrown up for you. So here are all of the things that have happened in 2017 with CRISPR technology. They removed HIV from mice. They edited a human embryo. They shrunk tumors, cancerous tumors in mice. They managed to generate semi-synthetic bacteria by adding brand new letters of genetic code. They were able to edit mosquito fertility, which helps with malaria. Remove Huntington's in mice. Huntington's disease is a terrible genetic disease if you're not familiar with it. It has lots of repeats, and the more repeats you have of the gene, the worse the disease becomes. They're also able to kill antibiotic resistant bacteria by using viruses, genetically modified viruses, in order to directly go in and attack this bacteria. They've also found that there's a potential use for biofuel through modified yeast, and there are now is a new regulatory body that's investigating CRISPR to make certain that it's safe for everyone. So these are all very, very, very good things that are happening in regards to CRISPR. So with 2017, I want to talk a little bit about some of these things. So in regards to the HIV that they removed from living mice, the important thing is that they survived. Now, some scientists in Pittsburgh were able to do this, and these mice also had tumors that were associated with the HIV, and they were able to shrink those, and they didn't spread any further. But it's good that all of these test animals survive. That's what's important. And now, okay, so I mentioned before about the human embryo being successfully genetically edited. There's a lot of ethical concerns in regards to that because they don't know exactly how this affects people down the road. But through the use of CRISPR, scientists were able to remove a gene that causes a very specific heart disease. Now, why is it easier in embryos than an entire person? Well, if it's in an embryo, you have less cells you have to work with. It's still developing, and you have a higher chance of it being completely eliminated as those cells divide and grow. If you're talking about an actual person that's already fully developed, it's a bit more complicated and a bit more difficult to reach system-wide in an entire human being as opposed to just a few tiny cells, right? All right, so they could use CRISPR to kind of slow down cancer growth. So what they did is they used CRISPR to try to target this command center of cancer that's responsible for excessive tumor growth. Now, if you're not familiar with cancer, you normally have two genes that are tumor suppressor genes, and if those are turned on and expressed, then that means that your cells will undergo what's called cellular death. They will die on their own because those tumor suppressor genes kick in. Well, various forms of cancer like to turn off those genes, and if those genes are off, then that means your cells are going to divide and divide and grow and grow and divide and divide and grow uncontrollably. All right, so scientists were able to create this cancer annihilating gene that shrunk tumor cells in mice. So that's pretty cool. So they're able to kind of shrink them in that regard and kind of initiate that apoptosis. All right, so in regards to generating new species, that's a potential that we could see. So scientists have been able to breed various types of bacteria with 6-letter genetic code, because we normally have 4-letter genetic code, G, C, A, and T. So they were able to introduce 2 new letters in order to generate this semi-synthetic organism. This has all kinds of different potentials, but the thing is, it's just kind of a cool thing that we know about right now. Now, in regards to some of our deadliest diseases, I had mentioned about mosquitoes. So if we're able to effectively edit the fertility genes in mosquitoes, we can limit them spreading malaria. And this is kind of a cool thing, is that since we now have CRISPR technology where you can edit multiple genes at the same time through that mutated version of Cas9, scientists are able to modify multiple genes at the same time, going all at once. And then I mentioned viruses that can kill antibiotic-resistant bacteria. So they're able to take bacteriophages. Remember I said that CRISPR-9, the CRISPR mechanism in general came from bacteria, and it was essentially, it's used to kind of remove viral genetic code out of a bacteria. Well, researchers are now using bacteria to go in and introduce genes to kind of get rid of that antibiotic resistant aspects to them. So that's kind of a new avenue that we can do, especially when we're dealing with more serious forms and strains of bacteria being resistant to antibiotics. DNA storage, you know, we could also, CRISPR can also revolutionize how DNA is stored. Which would be kind of cool. You know, their researchers are able to use DNA of live bacteria and store a GIF image using CRISPR, and then sequence the bacteria to kind of successfully reconstitute the file. So that's kind of interesting, but I really want to focus more on the biofuel. CRISPR has shown that you can genetically modify or genetically engineer a specific yeast strain to lead to sustainable types of biofuels. Okay, bye. I'll catch you later. People saying bye on the chat. So they were able to engineer certain algae that could also double the biofuel material as a natural counterpart. So we can use CRISPR not only in just gene editing for, you know, helping out with antibiotics and drugs and cancer, but also it has the potential to modify yeast and algae for biofuel. So this is all pretty interesting. And then now that there is a new regulatory body, the U.S. Defense Advanced Research Projects Agency, also known as DARPA, is now getting involved with CRISPR, and it's invested $65 million to a project called Safe Genes, where they want to ensure that there's no ethical misuse of CRISPR, and also that it is a safe type of technology to use down the road. But the thing is we have lots of things we have to overcome in regards to CRISPR before we can even do that. And so some of these things are, let me shrink myself a bit. There we go. Okay. Some of these things that we have to deal with before we can see this used in people. Efficiency issues. Oh, your daughter is a molecular gene major at OSU. She speaks of CRISPR often. It's exciting stuff to talk about. Okay, so issues with CRISPR. Efficiency issues. Actually attaching to the gene. Getting it to attach to the correct gene and not on the end. That's still kind of a bit of an issue, and it takes some time to overcome that. Another issue. Off-target DNA effects. So sometimes you have genes that look very similar to other genes, and you don't necessarily want to take out one gene that may be beneficial, but it looks very similar to the yucky one. What if it takes out the wrong one? You know, so we have some off-target DNA effects here that we got to work out, right? Now, delivery of the enzyme. All right, some people are like, how can this affect an entire body, you know, if you're trying to remove particular genes? Well, delivery of Cas9, that enzyme into tissues, presents its own problems. There could be a low entry into cells. You may not get as much into the cells that you'd like, which means you'd have to probably increase the amount. But then there's also issues associated with it degrading fast enough. And too much of it could be cytotoxic. So it can actually kill your cells if you have too much of it. And also you might have random gene insertion into your genetic code. And, you know, it may not be in the place that you want it to be. So there is an issue associated with that. Then you have something called genetic mosaicism. How is it going to reach all the cells? How is it going, is it just going to stay with this group? How is it going to spread to other cells? So you only have certain cells that have the fixed genes, and then you have other cells that don't have the fixed genes. This is what we call genetic mosaicism. And then lastly, I did mention the degradation of CAS9. Too much of it can cause a lot of problems. And so trying to, and humans require a large amount. So we've been removing Huntington's genes in mice, shrinking tumors in mice. But mice are like, you know, they're mice. They're teeny tiny. We're giant humans compared to them. So we would need a lot more of that CS9, and trying to get it to degrade in our bodies quickly and safely is an issue that has to be overcome. So while gene editing is really cool and all, we still have some things we got to get through, you know. Hello. So here is the last little image of a cool CRISPR thing going on. So I love this GIF image. It's just kind of cool. I do like GIF images. So the last little bit, while this is a bit more complicated, you know, while CRISPR is a bit more complicated than this, this ultimately kind of gives us the idea of how CRISPR works. It goes through, you start reading, you know, you get your genetic code going through, and your CAS goes into that, and it's like, okay, here's the gene that we need to get, and then it starts fitting it through, and then it, you know, pulls out the code that you don't want. It's very, very basic, and it's beautifully simplistic, and it's not that hard to understand as long as you're, you know, able to kind of grasp the idea that, you know, this really isn't a machine. This is a biomolecular machine. So it's all inside the body. But there, since it's inside the body, we have things we have to overcome. But it's still cool nonetheless, yeah? So this has been a pretty short episode compared to other episodes of the Science of with gene editing. This has been pretty fun. So I'm going to show you like all my sources and stuff here in just a second. I just love this little guy. He's like going through and he's like, is this the right one? Nope. I'm going to say, oh, here we go. We fixed it. Yay. Hello. But that has essentially been the science of gene editing. I have all my sources here. There's a whole lot of them. There's a whole lot of them. Right here. Woo! And what I'll do is I'll post my slides on my website, scientistmel.com, so where you can go and pop on and download my slides, and then you can go and read all there is to read about CRISPR. There's so much more information. Some of these are really taxing type of sciencey articles as far as like from journal like Nature and stuff, but then others are pretty easy to read and they can kind of direct you. And now that you have a bit better understanding of what certain things mean and how they work, this may not be so demanding to kind of actually sit through and kind of read. So I want to take a moment to like thank all my patrons. I need to add Patrick on here, and I have a new guy. I don't have his name on here, and I need to give him a holler because he recently sponsored me on my Patreon. And you can be a part of that too on patreon.com/scientistmel. I actually have an audio podcast called #HeyScientistMel. I'm updating that episode today. And I talk about all kinds of different things on that website, and all of my patrons get early access to all of my episodes of Hey Scientist Mel, and they get their own feed. So, I don't have their name right off. That makes me nuts. But anyway, I do need to add Patrick, and I think, oh God, can't remember the other. I think his name is Ed, but I'll give him a shout out at the end after this. But thank you to my patrons. They make what I do possible. Tony, Lauren, Jen, Carl, Melanie, Anthony, Paula, Daniel, Carrie, Keith, Zachary, Tony, Graham, Tristan, and Jennifer. You make what I do possible, and I'll be able to stream onto more sources and platforms very soon, and I'm very excited about that. You can also find me on scientistmel.com. You may be watching me on Periscope right now or YouTube, and I'm also on Facebook. So thank you so much for joining me here today. This has been so exciting, and I'm so glad I got to talk to you guys. You can always hit me up on Twitter. You can hit me up on Facebook. You can go to my website and send me an email there. And look to Twitter for my polls in regards to what you want to talk about. And I always add new topics in all the time. But this has been so much fun, guys. I hope you enjoyed it as much as I have. I got to learn all kinds of new different things with this one, and I get to learn with you. So I hope you guys have a wonderful, wonderful day. And I will see you around. Have a good day. Bye.